Like once again, mic is mute. We are continuing with the program right now, and the next speaker is going to be Goran Mausa, who is head of data mining lab from IRI Center, University of Rijeka, and he's going to talk to us about artificial intelligence-based design. And artificial intelligence-based design of short peptides. And basically, what I wanted to give you as an introduction is that discovering new peptides with desired properties based on their sequences is a challenging task, as they are part of a very large search space and the principle responsible for the target properties, and not yet fully understood. Our approach to overcome this obstacle is the application of search-based algorithms coupled with machine learning in search of de novo peptide sequences that exhibit antimicrobial activities. We also use soft computing. They also use soft computing techniques to find peptides and catalytic activities, to predict their predisposition towards self-assembly, and to estimate the antiviral activity of selected candidates. So basically, like you may heard, it will going to be very technical talk, and not totally adore these kind of talks. And I would like to say thank you and to invite Goran to join us.
Hello. Can you hear me?
Of course. Yes.
Okay. So, shall I start?
Of course.
Yes. Okay. Thank you for the nice introduction. So my name is Goran. I come from the Faculty of Engineering, University of Rijeka. As you mentioned, I'm the head of data mining lab at IRI Center. I'm also a member of another center, the Center for Advanced Computing and Modeling. But what I'm going to talk to you about today is my main research agenda that I work as a member of the SPET lab. So I will briefly give you the introduction into this field. I will describe some of the artificial intelligence techniques that we use, and hopefully it will not be too technical, as you mentioned. Let me just start here. Okay. So besides living in a world that is heavily affected by the COVID-19 virus, we also live in a world of data-intensive discovery. But how does that affect our everyday work, especially in research? Well, throughout the history of science, the foundations of evolving new theories relied primarily on experiments. Through experiments, we gather data, and this led to new theories. In the last decades, there was an increased usage of computing, like high-performance computing, some advanced computing models, and they also led to additional data which turned into theories. Nowadays, today's foundation turned to data itself, so we try to learn from data and make new theories solely on data. Data and huge quantities of it that we have gathered and are still gathering are becoming the central point in many research domains. In search of meaningful patterns, initially we turned to simulations. But in order to run simulations, mathematical models had to be known in advance. This means that we required the level of granularity and locality derived from the underlying physics of the problem that we are looking at. For example, here we have a model of some climate change model. So if we were to build such a model, we had to know all the physical laws of nature, transform them into mathematical models, and write software, and then run the simulations. Usually these are very demanding simulations which require a lot of computing power. Recently we turned to data and we started to observe phenomena for which no models exist, no laws of nature, nothing. For example, here we have a graph that is derived from data of internet connectivity, something we did not have 20 or 30 years ago. And we also have a similar graph that is something completely different, and that is protein interaction in yeast. So this approach, as opposed to the previous one, includes almost seemingly arbitrary connections between entities. So we are searching for patterns within the data itself and not taking into account any mathematical laws known before. The modern techniques applied in these challenging settings, when scientists need to grapple with theoretical problems that are analytically intractable, mainly fall under the umbrella we call soft computing. So soft computing, as the opposite of hard computing, is based on probabilistic modeling and uncertainty. Many of techniques that are within this area are inspired by nature. For example, we have neural networks, which are becoming very popular in recent decade, evolutionary algorithms like genetic algorithms, swarm intelligence, and so on. The term artificial intelligence has a very broad meaning, but we might say that soft computing techniques are a big portion of it. So here we have a set of non-traditional means for effectively handling difficulties like nonlinearity, instability, a lack of sufficient and precise information about the system. And when we use these algorithms, we may not always find the best possible solution, but we can come often with some approximate solutions that are robust. And most importantly, we come to these solutions in a reasonable amount of time. So machine learning is a niche in data science and a subset of artificial intelligence that computationally searches for patterns or irregularities in data. And here I'm going to talk to you about how we use them for peptides. So I did a brief search in PubMed repository. So in the last 20 years, when I searched for the terms peptides and machine learning or genetic algorithm in only title and abstract, I come up with around 8,000 scientific papers with a clearly increasing trend. So this is a very hot topic that is revolutionizing the way we do science. But what are peptides? Let's see a brief about that. So peptides are composition of amino acids. Amino acids are chemical compounds made of amino group, the NH2 part, carboxyl group, the COOH part, and a residue. Depending on the residue, we have 20 natural amino acids with different properties, which can be categorized as presented here. There are also other ways to divide them. More importantly, they are biology's expression language, and they present rich chemistry with 20 natural amino acids, which we use to form peptides. So depending on their sequence, with length up to 50 amino acids in a chain, they form various peptides. Sequences that are above 50 amino acids in length are considered to be proteins, but this is an arbitrary definition. If we consider only 20 natural amino acids for 50 positions, which is the maximum length, we come up with 20 to the power of 50 of possible permutations. In other words, they form a huge chemical space. Conveniently for us computational guys, amino acids also have single letter codes which we use often to represent them. One of our earliest projects in this field was published 2 years ago. We wanted to design peptide libraries, and these are mixtures of peptides which are synthesized simultaneously that could later be analyzed in a single run by mass spectrometry. To achieve that, we had to make sure there would be no overlap between these peptides in the mixture within a single library in terms of their mass. So here is an example of an input to our algorithm where we can say, okay, for each of the 5 positions in a short peptide that only has 5 amino acids, please consider these 6 amino acids for each position. If we were to put all these amino acids in the mixture, we would come up with 6 to the power of 5, which is 7,700 peptides. But here we can see on this graph that mass diversity would be close to 0. On the other hand, our algorithm proposes smaller solutions, which are around 30 to 80 peptides big libraries, so much smaller libraries, but their diversity is aims towards 100%. The maximally diversified libraries were further theoretically evaluated in terms of their composition and physicochemical properties. These properties will be of interest in our later work when we analyze whether we can predict the functionality of peptide solely on its properties. Our intention was to cover large sequence spaces and provide a controlled exploration of peptide chemical space, both theoretically and experimentally. And we did that in this paper that was published last year. So in discovery of active peptides, we turn to soft computing more and more. Although it may seem very convenient to avoid time-consuming experimentation and change it with much faster in silico design, that does not mean there are no challenges. One of them is the problem of data imbalance. So similar issues present in various application domains from, I don't know, fraud detection, rare disease detection, software defect prediction, or other areas where only a minority of the instances that we have is the, well, thing we are looking for. So we're looking for the fish that looks in the opposite direction than most of them. The discovery of peptides possessing high biological activity is very challenging due to the enormous diversity for which only a minority have the desired property. We can say that we are looking for a needle in a haystack. Although we may not expect to have more negative results in exploratory studies, the fact that negative results do not get published in research slow down our progress in data science. That is why sometimes we have to remind our collaborators and our counterparts to collect all the data they can get and not just the positive outcomes that they usually publish. This leads us to the project entitled Design of Short Catalytic Peptides and Peptide Assemblies, or DSC in short, which is founded by the Croatian Science Foundation and 2 supports from University of Rijeka. So, as little is known about the principles that govern this catalytic activity of short peptides at the sequence level, we want to combine machine learning with experimental validation to develop a more efficient and economical approach compared to the unguided experimental evaluation. We aim to discover patterns in existing data and accelerate the discovery of new catalytic peptides. Another project of ours is called SARS-CoV-2 supramolecular mimetics for discovery of peptides that induce viral entrapment, or microtrap for short. In this project, we want to design peptides that mimic virus structure from the spike protein, which especially the part that is responsible for binding with human cells. We want to analyze this sequence, try to find proteins that would mimic this functionality, and then later try to find another peptide which would bind and, in a way, entrap this part of the virus. Why peptides to fight COVID? Well, besides being the building blocks of life, peptides are of great interest in the biomedical field as they can act as safe therapeutic agents, and they are attractive due to low toxicity of their metabolites. So in this project, we also want to find whether we can establish a self-assembly propensity prediction model. So based on a known aggregation propensity scores for shorter peptides, we started developing a probabilistic model to estimate the score of a larger set. So from using a sliding window of with 1, 2, or 3, we want to use known data to predict self-propensity of larger sequences. Given a spike domain from the COVID-19, its sequence will be sliced into overlapping 3 peptides and will be taken from the existing database to build this prediction. So far, we replicated a single amino acid hotspot model, and this is this one, and started building our own models, which are based on sliding window of greater width, so with D peptides and 3 peptides in mind. Our initial results show that there are some distinct hotspots within peptide sequences which can be used to supplement these three techniques together. We also started working on machine learning-based evaluation of antiviral activity. So we collected 600 positive and 600 negative instances of antiviral peptides. We used a package written in Python to extract features, physicochemical features like size, mass, charge, instability index, and similar. And then we used, for example, a random forest algorithm to predict whether a peptide is antiviral or not. And we got pretty good results using only 17 features. We also did a similar experiment, but with a different property, and that is the antimicrobial property. So we're, again, taking positive and negative instances. This time we had to collect them from two different sources. We have around 10,000 of them. We used another package that allows us to extract 66 features this time, so much more complex, like Croatia properties, electrical properties, and other. And here we experimented with slightly different algorithms. And for example, the support vector machine, which in literature is the most commonly used machine learning algorithm, which surprised us for peptide research, was again very, very good. However, we didn't trust these results so much, so we said, okay, let's check that a little bit better. So we developed a strategy where we will build two models. One model will be based on the representation that uses features, which are previously mentioned, and another model that will use only sequence as its source of information. So we will only say which amino acids are at each position. When we built these two models, we got accuracy of around 90% for both of them. So again, very good with the training set. But then we said, okay, let's take one of the peptides from the training set. Let's assume its name is TEST, and his probability for being antimicrobial is 99%. We make random permutations of these letters and come up with new peptides for which the initial model, the first model, would again say 99% to be sure that they are antimicrobial for each of them, because it uses features, which are basically computing average numbers for these properties. Then we would give the same sequences to the second model, which doesn't know features but knows the sequence. And then our initial results show that not all of them can be considered antimicrobial, which is obviously expected, but we just demonstrated that we have to be very careful. Although we got 90% accuracy, that doesn't mean that we already have very good models. Another thing that we are doing is the usage of generative adversarial networks, or GAN for short. Usually these type of networks are used to create some, let's say, fake images. So based on random input noise, one part of the network, which is called the generator, would make fake figures, and the discriminator has the task to differentiate between real ones and fake ones. Real ones come from a dataset, and fake ones come from the generator. As this model grows and learns, then the generator gets better and better at creating fake images, and discriminator gets better and better at realizing which ones are fake. So we applied this same principle in our project, and we say, okay, let's use noise. Let's make some noise to make random peptides. And these peptides are going to be compared to the ones that we have in our database, for example, antimicrobial. And then we hope to make the model find new peptides which would potentially have the same or greater activity that we are looking for. Currently, we are struggling with this model, so this is work in progress, because our generator seems to have some model decay, because it seems to train a rigid form from which generates only one type of sequence after a training batch. So although it has random noise on the input, its output is more or less the same every time. So in conclusion, traditional methods for discovering and synthesizing peptides are expensive and time-consuming, often involving months or years of guesswork and failure. We want to learn from this failure, we want to extract data from this failure, and then we want to make our models better. And instead of randomly guessing where should we look for our patterns, this is the way human thinks. So he finds a good solution and gradually looks for a better one. But with looking without any prejudice, without having any human-like restrictions in the way we think, we want to make the computer find this maybe even better solutions than the ones that we can come up with. Soft computing offers insight into complex relationships such as sequence function, which I mentioned. And this relationship is so far unknown and from the theoretical point of view. So basically, that's the best thing we can do. Finally, we may benefit from computational methods in a seemingly unexpected way, because this unbiased search that is free of prejudice assumptions may ultimately find superior solutions. And in the end, I would like to thank my collaborators, especially Daniela Kalafatovic from the Department of Technology, who is the principal investigator from this chemical part of this project. Also Ana Pina from Lisbon, Marko Perčić from my faculty of engineering, and our PhD students and other students who are helping us with all of these research activities. And our funding bodies, Croatian Science Foundation and University of Rijeka, for their support in financial aspect. And thank you for your attention. I think you are muted again.
Yeah, thank you. So I hope that next time I will remember to first time you then start talking. Okay. Yes. Basically, first of all, I would like to thank you for your time. I would like to also say that it was very interesting to listen to this. So basically, we have a few questions. I would like to start with the first one. And first one is, how much did you use swarm intelligence algorithms? Can you share with us some materials after the conference finish, which are reliable to the CI?
Okay, so swarm intelligence. Well, so far we didn't use swarm intelligence. We use genetic algorithms more. So basically, they do the same thing. They're used for optimization purposes. In our experimentation, we used genetic algorithms, well, because we had a very clear representation and our search space. Well, swarm intelligence, if we think about, I don't know, particle swarm optimization or other algorithms, maybe have some assumptions about the search space for which we didn't want any implementation, because our main representation of the search space is a binary one. So we're basically transforming the search space into a bit string and then looking for the optimal subset. So yeah, so far we haven't used swarm intelligence, but genetic algorithms more.
Okay, the second question is, how come peptides are such a hot topic? Why did you say that this is a revolutionizing science, and in which sense?
Okay, so when I said it's revolutionizing science, it's revolutionizing science related to peptide research.
Okay.
So not in this broad aspect of science itself. Well, they are a hot topic because from the biological point of view, peptides are very simple to replicate, to build, to produce. They may be organized in different ways in a very simple way, comparing to much more complex structures like proteins. Proteins, well, we have them in our bodies and elsewhere in the nature, but these ones are the ones we can produce. And we often find peptides which are safer, let's say, from the biological point of view, as therapeutic agents. So they're used to cross the blood-brain barrier. They're using to, I don't know, deliver some drugs or something like that. So they are used for various purposes, and that's why they're a hot topic, because they have very, very wide range of activities. So besides antimicrobial and antiviral, which I mentioned, there are also anti-cancer peptides, antifungal peptides, or other.
Very interesting, to be honest. Like, we have two related questions to this one. I'll try to squeeze it all in 2 minutes. So if you can just give short answers. What are the connection between the Pfizer vaccine methodology and peptides? Is there any? If no, what is the difference?
Okay, I have no idea. So, my part of the work here is mainly in the computational part and the artificial intelligence. So I would have to ask this my chemists in the group if they know the answer to that question.
Maybe we can do a short follow-up maybe on a video, so we can post it for this. I will send to you this question. Maybe you can just record 2 or 3 minutes video to explain to the people. I think this is very interesting because this people can relate to this.
Yeah.
Okay, and the fourth and last one is, can you elaborate a bit more? What is the difference between 82% and 92% F1 score in the prediction of let's say antimicrobial peptides? What does this mean in the practical sense of technology development? Not only like statistics, but how much is this important to get better results? And after that, I think that you talk that you get even better results in this one. So just so people can understand what is the game changer here.
Yeah. So basically, well, the difference between 82% and 92% is, well, relatively big. It's 10%. Yeah, but that doesn't make much difference from the experimental point of view. So in an experimental point of view, one way to do the experiments is without any support from the computational algorithms, such as, let's say, support vector machine or others that we used. And then you don't have any percentage. So you're just making random guesses. You're making, most of the time, researchers make small changes to the sequences that are known to be active, for example. On the other hand, here we make big changes, random changes, but these changes have some support in the algorithms. In the end, even for something that we are sure 92% that has some, I don't know, antimicrobial activity, in the end doesn't have to have antimicrobial activity, but the probability is greater than if we don't have any such decision support system, let's say.
Okay. So basically, with this one, we are finishing with talk. Goran, I would like to personally thank you, and as well University of Rijeka. And I would just like to say that we are starting a strategic partnership with the University of Rijeka. They are doing a lot of excellent stuff. From an academical point of view, we're very glad that we are having you in our partners pool regarding like development of AI and in the whole region. I would also like to all the times say thank you to Sandra, who was the first one from Rijeka who come to Belgrade, and she created a part of the conference, Data Science Summit we're going to have this year. But not to take any more time, we can switch to the next speaker. It's all going to be Mate Pitsukaric, and he's from Atomic Intelligence, and he's going to talk how AI is transforming AI music industry. Okay, so we are coming back very shortly. Thank you once again.
Bye. Thank you.